Hello, everyone. Here I'm going to give a talk titled as CRISPR-Enabled Gene Regulation Tools for Biotechnology Applications. I'm Baojun Wang from the College of Chemical and Biological Engineering of Zhejiang University. I'll start by giving an overview of the research in my lab. In brief, we're working at the interface of biology and engineering. We develop enabling tools for synthetic biology and building customized genetic circuits for sensing and computing of multiple cellular and environmental signals with application ranging from biosensing, biomanufacturing to biotherapy. Our research comprises both foundational and applied synthetic biology. First, we design synthetic genetic circuits, such as this layered digital genetic logic gates, and also develop synthetic biology enabling tools. For example, recently we have developed eukaryote-like CRISPR-enabled gene activation tool in bacteria, and also a CRISPR-based RNA sensing tool in bacteria. And we also engineered an expanded library of orthogonal split engines that allows for scaled assembly of multiple peptides. And next, we apply these circuits and tools to develop various applications, including synthetic cellular sensors for environment and health, and engineering bacteriophages to carry sequence-specific nucleases to precise target gut pathogens. In addition, we engineer these benign bacterial cells to specifically regulate and care cancer cells. Our work is supported by leading industrial partners in the field, including Microsoft for biocomputing, Huawei for biomaterials, and US Navy for biosensing. The CRISPR systems, we know that it originates from adaptive immune system of bacteria, and CRISPR stands for clustered regularly interspaced short palindromic repeats. The CRISPR loci are clustered of short DNA repeats and separated by equally short spatial sequences of phage and plasmid origin. And this repeat and space sequence is flanked by operon of CRISPR-associated genes that encodes the machinery for the immunization and immunity stages of the systems. And in the immunization stage, the space sequence are captured upon entry of the foreign DNA into the cell and integrated into the first position of the CRISPR array. And in the immunity stage, the spacer is used to target invading DNA that carry a cognitive sequence for destruction. The spacers are transcribed and processed into small CRISPR RNAs in the CRISPR RNA biogenesis phase. And these small RNA act as antisense guides for Cas RNA-guided nucleases that locate and cleave the target sequence in the invader's genomes during the targeting phase. Endonucleases, if an artificial guide RNA can be produced to guide the Cas, for example, the Cas9 nuclease to target double-stranded DNA and cleave at a site that pairs with 20 base pair spacer sequences of the guide RNA, and also have adjacent PAM sequences. Due to its simplicity and ease of design, this sequence-specific RNA-guided endonuclease can be used widely as a gene editing tool in different cell hosts across all the three kingdoms of life, including bacterial, and animal, and human cells. The catalytic and inactive, the date Cas9 showed as deep Cas9, in which the point mutations have abolished the DNA cleavage by the HNH and RUBC domains of the Cas9 protein, can be repurposed as a transcription activator, showed as CRISPR-A, or repressor as CRISPR-I. And for CRISPR-A, dCAS is fused to a transcriptional activator as a single guide RNA programs dCAS fusion to activate the transcription of the target promoter. A commonly used CRISPR system in mammalian cells uses the VP64 domain, while for the CRISPR-I, the dCAS is fused to a transcriptional repressor. A common CRISPR-I system in mammalian cells using the CRAP domain to achieve this. And the dCAS CRAP can direct the heterochromatin formation at the promoter targeted by the guide RNA and reduce target gene transcription. And here is the outline of my talk, which comprises 3 parts. First, I will describe the design principles and biotech applications of a eukaryote-like CRISPR-enabled gene activation tool in bacteria. And second, I will discuss how reprogram the transacting CRISPR in type 2 CRISPR systems allows repurposing RNAs as CRISPR RNAs and sequence-specific RNAs by senses. The third, I will describe the engineering of the P1 bacterial phage meat to deliver CRISPR-Cas9 antimicrobials against the Shigella flexury, which is a barrier causing gut pathogen. The CRISPR activation is a type of programmable gene regulation which has several advantages compared with traditional various gene activation devices. First, the guide RNA-based approach can specifically regulate and binding, provides very high orthogonality between the parallel devices and the circuits with their genetic background. And second, the programmable principle supports large library of artificial promoters which have uniform activation mechanism. And this means that they have a similar dynamic behavior and quantitative feature. And for both of the Onion-2 CRISPR system in bacteria before, based on sigma-sensitive systems and CRISPR complexes, and leads to target upstreams a region close to the core promoter sequences. The available region is short. And in addition, there's low universal block mechanism for sigma-sensitive promoters, so basal output at off state is always significant. While for sigma-34 systems in E. coli, the flexible loop structure allowed the promoter tolerating a wide target range at the upstream UAS region. The original block function of the sigma-34 factor, which need to be unlocked by the ATP-dependent activator, could achieve low basal output at off state, which also could improve its dynamic range. And to address the limitations of the previous CRISPR systems based on sigma-sensitive promoters, we designed our systems based on sigma-34 dependent promoters. On the other hand, the specific eukaryote-like mechanisms also provides advantage for engineering sigma-34 activation systems as programmable regulation tool for genetic engineering. We designed our CRISPR-A tool based on the sigma-54 dependent promoters. It works by directing the dCAS line to the promoter upstream activated sequence region with a guide RNA design with extra optimal domain of an RNA binding protein to recruit the fused sigma-54 activators to the promoter for transcriptional activation. In contrast to sigma-sensitive promoter system, the flexible loop structure in our CRISPR-A allows the promoter tolerating wide dynamic range at the upstream US region, in addition to have lower basal output compared to due to its low spontaneous transcription initiation. For example, we can by doing this, we can achieve dynamic range more than 1,000-fold, which is compared to previously the dynamic range of less than 10-fold. First, we demonstrate our CRISPR systems can activate various wild-type sigma-54 dependent promoters in E. coli bacteria with very high dynamic range. For example, it can activate the PSPA promoter in E. coli, which is related to the phage shock stress response of E. coli, and also the HRBL promoter, which is related to the hypersensitive response of the plant pathogen Pseudomonas syringae. Our CRISPR system can also activate the wild-type NIF-H and NIF-J promoters within the nitrogen fixation pathway from Klebsiella oxytocin in E. coli. If we choose the proper guide RNA with a suitable PAM site in front of the two promoters. And this slide shows that our CRISPR systems can function in non-model bacteria with biotechnology relevance. We demonstrate by using the CRISPR-A to activate the sigma-54-dependent nitrogen fixation pathway genes in Klebsiella oxytocin, a free-living soil bacterium that can convert the atmospheric nitrogen into the nitrogen fertilizer ammonium. On the lateral context, this pathway is strictly locked on aerobic conditions. However, our CRISPR tool can bypass this host-regulated inhibition to activate the native NIF-H and NIF-J promoters for nitrogenase expression in this pathway, as shown by the RT-PCR data on the bottom right panel here. And this CRISPR tool should act as a very powerful tool for nitrogen fixation research and biotech applications in the future. In the next two slides, I will demonstrate the application of this CRISPR tool as a reusable profile scanning platform for metabolic pathways. Our systems comprise of two parts. The first is a reusable CRISPR guide RNA generating plasmid library comprising the pre-coded gene expression profiles. The DX, Cas9, and activated proteins are also reusable, and the expression levels can be tuned with external inducers to control a pathway's global expression strengths. The second, the target pathway only needs to be assembled once with designed orthogonal sigma-54 promoters for each pathway genes. And by doing so, our CRISPR tool can project diverse transcription profiles carried on the multi-guide RNA generated plasmids onto the same multi-gene pathway, which then can be screened to identify transcription activation patterns with maximal production. We demonstrated its proof of concept using the violence production pathway comprising 5 genes in E. coli bacteria. Notably, violensing is a valuable anti-cancer drug with a visible purple color. Here, the guide RNA generated library was synthesized with 3 constitutive promoters of different strains to control each cochlear guide RNA for activating the VIO-A, VIO-D, and VIO-C genes in the pathway. So together, there will be 27 combinations of different activation patterns. As shown in the middle left panel, our CRISPR system, which is this generated library, can produce cell colonies of diverse production profiles in contrast to the controls with either low guide RNA generator or a generator with all strong promoters. The representative 4 colonies corresponding to the weak, intermediate, and strong production were then sequenced to compare the pathway transcriptional activation patterns. On the right panel, it shows that expression level of the VIO-A gene in red dots here is vital to the violence production level in this pathway. In summary, we engineered a novel CRISPR activation system that enables programmable eukaryote-like gene activation in bacteria. To address the limitations of previous CRISPR system based on sigma-70 promoters, we designed our system based on the sigma-54-dependent promoters and have achieved a programmable eukaryote-like bacterial CRISPR activation system with ultra-high dynamic range. It facilitates orthogonal, multiplex, and multi-layer gene regulation. It functions in non-model bacteria with biotech relevance. And finally, we demonstrate its application as a reusable profile scanning platform to rapidly identify transcription activation patterns for maximal production in multiple gene pathways. In the second part of this talk, I will discuss the programmability of CRISPR-RNA and transacting CRISPR-RNA hybridization in type 2 CRISPR system, and how the reprogrammed transacting CRISPR-RNAs allows repurposing of various RNA as CRISPR-RNA and act as sequence-specific biosensors. We know that the type 2 CRISPR-Cas systems, the guide RNA consists of CRISPR RNA and transacting CRISPR guide RNAs, as this graph shows here. This is the two guide RNAs, and together with the double-stranded DNA forms a triplex complex. So in the middle, it's a CRISPR guide RNA, and the yellow region is the 20 base pair special sequences, and the red region is the repeat region that pairs with the transacting CRISPR RNA. We know that in the space region, the 20 pair space region is programmable and allows you to target in the double-stranded DNA. And then we ask question, can the hybridized sequence of CRISPR RNA and transacting CRISPR RNA, the red region, if it can be also be programmed? And if this is the true, and then we can ask, can we use the reprogrammed transacting CRISPR RNA that in this way we can repurpose any RNA as CRISPR RNA. In that way, it means the whole CRISPR RNA molecules will be programmable. So this is the question that we and also the hypothesis we start from. So to investigate these questions, we engineered our the single guide RNA CRISPR system we have introduced just before, and we make it as a two guide RNA CRISPR activating devices. By separating the single guide RNA into two guide RNAs that pairs in the repeat anti-repeat region, and we also remove the aptamer regions for recruiting the activators to the end of these guide RNAs. So to demonstrate that if the hybridization regions of the CRISPR RNA and CRISPR transacting CRISPR is programmable, we mutated the 9 base pairs during these hybridized regions of the two guide RNAs, and you can see from the data here, all the mutations are, all the mutations in the line best pair of matching regions. It also allows the CRISPR devices to activate at high efficiencies, and also you can see that when you mutate the base pairs close to the space frequency, it can support very high orthogonality even at a single base pair resolution. Now this demonstrates that the CRISPR RNA and transacting CRISPR RNA hybridization is programmable, and this allows that we can reprogram it to also to achieve these programmable activations. And next, we want to demonstrate that if we can use the reprogrammed transacting CRISPR RNA based CRISPR reporting circuit to report the presence of specific mRNAs of some gene expression. In this case, we like to monitor the mRNA of the red fluorescent protein that produced from the AHL small molecule induced promoter here. So we designed use the CRISPR devices by reprogramming the transacting CRISPR RNA that can pair with the MRFP of the RFP here. If it conforms this, compares with conform this complex with the decast line, so you can activate the CRISPR-A promoter to produce a green fluorescent protein reporter. We choose 3 sites of the mRNA of the RFP here. You can see here 2 sides of it. The R2 and 3 can give very good outputs, and this demonstrates that we can hijacking the mRNA of red fluorescent proteins as the CRISPR RNA and to activate our CRISPR output. And you also can see that this the hijacking or detecting of mRNA did not affect the production of the red fluorescent protein, and also the levels of the red fluorescent protein and the red-green fluorescent protein, they are in they shows a linear relationship. Means we can also quantitatively monitor the mRNA levels of the red fluorescent protein and use this reprogrammed trans-acting RNA-mediated CRISPR-A circuit. Next, we would like to see if we can also monitor endogenous RNA produced from the cell genomes and use our reprogrammed trans-acting CRISPR RNA-mediated CRISPR-A circuit. So here we choose the arsenic-responsive pathways in the E. coli genomes. This is comprised of the arsenic-responsive operon. We can produce this polycistronic mRNA in responsive to sodium arsenide added to the medium. And similarly, we designed a reprogrammed trans-acting CRISPR RNA that can recognize a region on this mRNA of these arsenic-responsive operons. And as you can see here, with the increase of the sodium arsenide in the mediums, our CRISPR devices also can generate fluorescence in proportional to the arsenic levels added here. And you can compare to the wild type here. And we also can increase the dynamic by using shorter region of the repeat and anti-repeat regions in these CRISPR devices here. And this demonstrates that we can reprogram the trans-acting CRISPR RNA and to repurpose the endogenous mRNA as CRISPR RNA in vivo and also activate this CRISPR reporting circuit. And this also shows that in the future, we can use this mechanism to monitor the transcriptional activities of various environmental response promoters on the genome. So next, we also want to develop. We also develop a method that can monitor these environmental RNAs in vitro. So here we designed a circuit in the cell-free systems. And this is a DNA molecule that can generate this. Default can generate this locked broccoli RNA molecule. But in the presence of the target RNA molecules, our reprogrammed trans-acting RNA can pair with it, and together with Cas9, it can cleave this reporting DNA molecule circuits. In that, by doing this, this cleaved DNA molecule will produce a functional broccoli RNA, which can unlock this broccoli RNA structure that can produce fluorescence in the presence of the fluorescent dyes. By doing this, our circuit can report in the presence of the SARS-CoV-2 RNA molecules within 10 minutes, as that's shown here, and also the output outcome, which is generated the fluorescence that can be imaged by a full camera, and when the fluorescence accumulates to high levels with time. We named this approach as the atypical RNA activated transcription halting alarm systems, short as ACASA, in memory of Agatha Christie, which is the best-selling novelist in the world, that known for his popular detective novels. So in the last part of my talk, I will describe the engineering of the bacteriophage P1 based phage meat for the delivery of CRISPR-Cas9 antimicrobials to achieve selective killing of diarrhea-causing gut pathogens such as Shigella factory. Our motivation lies in that diarrhea is the second leading cause of deaths in children under 5, killing around 800,000 per year. It is also a leading cause of malnutrition in children, predominantly in Africa and Southeast Asia. And traditionally, antibiotics can be used to treat disease. However, this can worsen the children's malnutrition status by destroying the gut microbiota, in addition to aid the spread of antibiotic resistance issue. Gates Foundation were looking for alternatives to address this challenge. We proposed to engineer the broad host range lysogenic bacteriophages comprising strain sequence-specific nucleases to cleave the chromosomal DNA of target gut bacteria, hence achieving highly selective killing without destroying the commensal bacteria. To achieve this, we used the bacteriophage P1 as the delivery vehicle, which is a broad-spectrum lysogenic coli phage that can infect 25 species of gram-negative bacteria. On left, the synthetic P1 phage meat are constructed to carry the CRISPR nucleases and gut RNAs and produced in the production and cells that already contain the lysogenized the phages. On the right, the resulting phage meat particles can then infect the gut bacteria to deliver nuclease to cleave either the chromosomal DNA or the virulence vectors within the target pathogen, such as the Shigella factory, the enterotoxinic H. coli, etc. We validated the engineered P1 phage meat therapeutic efficacy both in vitro and in vivo. Here the data shows that the P1 delivered CRISPR-Cas9 infector is capable of improving outcomes in the zebrafish embryo model of bacterial infection. The left panel shows that the Cas9 phage meat can significantly reduce the bacterial load in vivo compared to the empty phage meat control. While the right panel shows that the Cas9 phage meat can significantly promote host survival of the zebrafish embryo larvae infected with the lethal challenging of Shigella factory bacteria. Next, we show that we can expand the host range of phage meat by engineering its tail fiber proteins in order to deliver Cas9 antimicrobials into new clinical relevant human gut pathogens, including Shigella factory and Escherichia coli 0157H7. And first, we developed bacteriophage P4 cosmate systems for the delivery of the Cas9 antimicrobial. The genetic engineering of the phage tail fiber improves the transduction efficiency of the cosmate DNA in Shigella factory M90T, as well as allows regulation of a long-native host, E. coli 0157H7. We also show that the transducing units with the chimeric tails enhance the overall Cas9-mediated killing of both these pathogens shown here. The key message I would like to convey here is that synthetic biology enables programmable living cells for new paradigms computing, sensing, and manufacturing to address some of the real-world challenges and needs. For example, cells can be programmed to detect various disease signals, including cancers, or as low-cost sensors for environmental pollutants and pathogens, or act as smart living drugs in the human body. And in summary, here I have presented 3 short stories, including development and the biotech applications of 3 CRISPR-enabled tools. And that is a eukaryote-like CRISPR gene activation tool in bacteria, and reprogramming the trans-acting CRISPR RNA to repurpose various RNAs as CRISPR RNAs or as acting as sequence-specific RNA biosensors. And the third, we can engineering the broad host range P1 fatty mate to deliver Cas9 nuclease for selective killing of the gut pathogens. And all these cases have shown that the CRISPR-enabled gene regulation is a powerful and versatile tool to achieve these synthetic biology ambitions. Here comes the end of my talk. I would like to thank my team who have done the work, in particularly Yang Niu and Yang Wei Huang here for the work I present here today, and my collaborators in both academia and industry for their invaluable support. And lastly, thanks to various funders. With that, I thank you all for your attention.